I am Dr. Andrew Shoebridge, so I'm an early career researcher from the South Australian Health and Medical Research Institute, and I'm one of the facilitators for today's event. But firstly, I would like to acknowledge the traditional owners and the custodianship of the land on which this workshop is held, and the land in which our attendees are joining from across the country, and pay our respects to the elders past, present, and emerging. So this webinar today has been organised by the Australian Dementia Network Early to Mid-Career Researcher Accelerator Group. And our aim is to support and advocate for early and mid-career Australian dementia researchers, and we help develop future research leaders. As a group, we are co-chaired by Associate Professor Nadika Dissanayake from the University of Queensland and Dr. Stephanie Wong from Flinders University. So on behalf of the ADNeT Early to Mid-Career Researcher Group, I would like to formally welcome everyone to this workshop on overcoming translational research challenges. So translational research, as you might know, has many challenges, including identifying feasible studies, maximising grant success, and working alongside clinicians to develop interventions. The aim of today's webinar is just to have a discussion with a panel of some successful neuroscience researchers and clinicians on their experiences with translational research and offering any tips they may have to our young researchers. Please note that the webinar is being recorded today, and this will be made available on the ADNeT webpage. So I'm joined today by my co-facilitators, Dr. Celia Furia of SAMRI, Dr. Jackie Wesson of the University of Sydney, and Dr. Suraj Santani of UNSW. Now, our webinar is fortunate to feature 3 expert panelists today who are experienced in the translational research pathway. We have Professor Clement Loy of Macquarie University, Dr. Sarah Hellawell of Curtin University, and Associate Professor Cedric Barty of SAMRI. We will hear from each of the speakers about their personal experiences and journeys first, and then we will open up the floor for some Q&A. And throughout the session, please type any questions you might have into the Q&A panel on the right-hand side of your screen, and we will be able to get to those later. So I would hand over to our first speaker, but I will take over that introduction on behalf of Celia, who is absent. So I would now introduce our first speaker, Professor Clement Loy. So Professor Clement Loy is a clinical epidemiologist and cognitive neurologist with a research interest in molecular genetics. He is director of the Huntington's Disease Service at Westmead, which serves about 75% of people with Huntington's disease in New South Wales. He's an investigator in a number of NHMRC and NIH-funded projects and clinical trials for people with Huntington's disease. As the newly appointed head of the Macquarie Medical School and Dean of Medicine for the Faculty of Medicine, Health and Human Sciences, he continually strives to translate evidence into practice and serves on a number of governmental advisory panels, including the Economic Subcommittee of the Pharmaceutical Benefits Advisory Committee. So I'll now hand over Clement, if you could please just share with us for a few minutes of your own journey and experiences. Thank you, Clement.
Thanks very much for the invitation, Andrew and Edwin. Yeah, I mean, it's a nice opportunity to also think through my own career. There are many academics with a far more focused and stellar career than I have, and my career has developed through a rather circuitous sort of route. And this is largely curiosity-driven, but I'm very, very lucky to have a very satisfying career. And I think at the end of the next couple of minutes, I will summarize some of the lessons I've learned, but I'll just briefly tell you how my career has developed. So when I left school, I was really wanting to be a mathematician rather than a doctor. But by the 5th year in my 6th year of medical degree, it finally makes sense to see patients. And at that time, there was a psychiatrist, his name was John Snowden. He introduced me to a patient with Pick's disease, which a lot of you would know as FTD now. And that really piqued my interest, sorry for the pun. And that got me interested in caring for people with dementia. Now, in medical school, you can also do an option term. And at that time, I was still thinking about doing mathematics. So I did a clinical epidemiological project with Les Erwick, who ended up being a really long-term mentor of mine as well. So when I graduated and started working as an intern, because I knew less, I did a master in clinical epidemiology. Evidence-based medicine was quite new then, and was quite unusual for people to do that. But that actually gave me the opportunity to learn about skills in statistical methods and in study design that has stand me in good stead for many years to come. At that time, I wanted to work in dementia. And most dementia doctors at the time in Sydney were psychiatrists. But unfortunately, I couldn't find a psychiatry training position at the hospital I was at. So I went into neurology instead. And this is, one of the many times in my life that things didn't quite go as planned, but actually worked out well. So in the middle of my physician training and after completing my initial exams, Les, my mentor, said to me, look, I've got a part-time job teaching in epidemiology. So I actually took a year off and did that. And people were asking me, why did you interrupt your clinical training to do something different? But that ended up being a really important year for me. I learned about teaching, about how universities work, and actually ended up being a 20-year association with that program, and I ended up running it sometime later. The teaching position was only half-time, so I thought I would love to learn something new, and at that time, and I'm quite old, so at that time, molecular biology is new, so I volunteered and did some work in a molecular genetics lab with Peter Schofield at Garvin at the time, and that eventually also led to more. Then I finished my neurology training. I still want to do dementia, but there was no training in Australia for that at the time. In fact, a lot of people said to me, look, dementia is not neurology. Why are you training in that? But I was lucky that there were some options in Queen Square, London and San Francisco, and I went to Queen Square. Then serendipitously, again, between my Sydney job and the Queen Square job, I've got 6 months, a bit of a gap. A friend of mine said, look, we need someone working in Huntington disease for a little while. Can you help out? So, I spent 2 days a week for 6 months working in that unit and with Liz McCusker. And in the end, this unplanned pit stop also ended up being, highly influential, and it's something that I'm working in now. So going overseas was great. I think, as a friend of mine said, you're most likely to learn something you can't learn locally. But even if you didn't, people thought you had. And in my case, I certainly learned a lot that I couldn't have learned here. And unexpectedly, I learned about inborn errors of metabolism, which I could never have trained in there. But after 18 months, they asked me to stay for another PhD, for a PhD. And but it was too cold and too dark, and I wanted to come home. So I came back and worked with Peter Schofield in the lab instead. And that's the person that I just happened to be working with before I left. So at that time, there were only 3 other neurologists in Sydney with cognitive training. So as I did my PhD, I ran a clinic in parallel, which built up a cohort. And interestingly, I ended up working on the DNA of the patients that got me interested in dementia in the first place. So it's really nice. As I was finishing my PhD, Liz McCusker, that I worked with briefly before I left, said, look, do you want to come and join and lead the service? Again, that wasn't what I had in mind. I thought I'll run dementia genetics as my theme. But then I realized that as an epidemiologist, Huntington disease is actually much more common than all of those put together. And there'll be a lot more scope in the future in terms of human studies. So I took that on. And concurrently, what I was doing before with clinical epidemiology also has a job that came up. So I took on a full-time, a half-time research and teaching position at Sydney Uni. I know a lot of researchers, when you start off, you say, look, I want to get fellowships and, I want to concentrate on research. I don't want to teach. But, that job has been fantastic for me. It's been great for family. When the kids were young, it gives you opportunities to not have to worry about, your salary for the next 3 years. It also, and within that, I had a few fellowships, which gives you relief from teaching and clinical work. So it really worked out well. So if you do see one, think about them. So when I started my first definitive job, I tried to bring my 2 jobs together, Huntington disease and clinical epidemiology. So we started with a prevalence study and an observational study. And at that time, there started to be trials in neurology, and I joined as a site investigator for one of the academic trials from the US. And after learning about that, we started doing commercial trials and eventually designed and obtained funding to run 3 trials of our own. And 2 of my MRFF grants right now is based on that. So, I mean, for one thing that's particularly satisfying for me is when we were in the lab, we use RNAi and ASO to manipulate your cell cultures. And now we're putting that in people to see if it helps them. So it really has been a very fortunate time for me. And my background, which is very odd in basic sciences, epidemiology, and clinical neurology, also enable us and myself to work as a member of the Pharmaceutical Benefits Advisory Committee, which is very satisfying work. Then about 5 years ago, I started thinking, look, you know, one of the challenges in running a service is that you need to understand how to manage teams and how to understand financial statements, and started doing an MBA. It took a long time, and my wife wasn't terribly happy about it. But I'm kind of a curiosity and learning driven person. So it was actually quite fun. You just watch a bit less Netflix or Disney or whatever you do. So that sort of became part of what I do. I started a management job at the hospital, and this week I just started being Dean of Medicine at Macquarie. What I'll be doing 10 years time, I'm not sure. You can ask me then. So that's a really long way of describing what I do. So what have I learned? I think first, if you're curious about something, and don't be afraid to learn it, even though it doesn't seem fashionable at the time. And in fact, you know, things that are not fashionable means that there's less people know about it. When it becomes important, you're in a better position. Second, if you're curious about a lot of things like I am, try to bring them together somehow. So if you're interested in a lot of different methods, then try to apply it to one area of content area. If you're interested in a lot of content area, then try to be the expert in one research method. You know, people, when they look for collaborators, they always look for the go-to person for this method or go-to person in this content area. And finally, don't be afraid of detours. I think, you know, most of my detours that were gap between formal things actually became career shaping for me. You know, in terms of translation, I'm lucky to have, you know, exposure to all these things, and I'm really more a jack of a few trades. And, you know, that allows me to collaborate quite intelligently with people. And I think that's some of the lessons I've learned. So I hope that wasn't too boring, and yeah, look forward to any questions. Thanks, Andrew.
Thank you so much, Clement. No, I really appreciate it. That was very insightful. Yeah, looking forward to asking you some more questions. It sounds like you've got a wealth of knowledge behind you. All right, so now I'll just hand over to Jackie, and she'll introduce our next speaker.
Thanks, Andrew. Hi, I'm Jackie Wesson. I'm a senior lecturer in occupational therapy at the University of Sydney. I'd like to introduce our second speaker today, Dr. Sarah Helliwell. Sarah is a research fellow at the Perrin Institute and Curtin University with a career focus on traumatic brain injury. Her research incorporates both clinical and preclinical research programs spanning the spectrum of brain severity, enabling her to rapidly identify clinical problems and translate bedside to bench and back again. Dr. Helliwell's current clinical neuroimaging research focuses on the development and implementation of objective tools to quantify and monitor progression of brain pathology in concussion. Thank you, Sarah.
Thank you, Jackie, and thank you so much for inviting me to be here today. It's really exciting to learn more about the network and to see the excellent work you guys are doing in promoting EMCR careers and providing information as well. It's really great. So I think I am in the sort of unlucky position to be speaking after Clement, who is so incredibly impressive. But I think he had a really excellent point that often careers driven by curiosity end up being particularly rewarding in that you're not going on a particularly linear path, but you're really doing what you're passionate about, and you can end up in quite unexpected places. And that's definitely been the case for me. So I started off in my PhD doing basic research on traumatic brain injury, which, as most of you will probably know, there's no treatment for, and long-term, many people who suffer a traumatic brain injury end up developing cognitive and brain impairments, and end up with dementias and Parkinson's and things like that. So during my PhD, I was working on a therapeutic preclinical trial for erythropoietin, which, if you don't know EPO, it's essentially the drug that the cyclists use for their doping. That's the Lance Armstrong drug. And it's a really excellent drug because it is a multifunctional agent. So it has a lot of different modes of efficacy in the body, but its main job is to boost red blood cell production, which is why it's really good if you're looking to be a sports cheat. But we thought that it could be, there'd been some trials showing that it could work in the preclinical space, at least, in reducing some of the pathology and improving long-term outcomes. So I tried this drug in my animal study, but at the same time, I was also doing behavioral tests and taking blood so I could monitor whether this drug was having any efficacy in real time, and looking at both the levels of the drug within the blood over time and looking at certain biomarkers that we were only using sort of in the preclinical space to sort of use it as a theranostic monitor of progression. So I was really curious at the same time, or slightly after I think I'd started using this drug in my PhD, I was working at Monash University, but physically on the campus of the Alfred Hospital in Melbourne. And we had led at the Alfred, but an international study using this drug in human patients with severe brain injury, which is what I was also studying. And I, as I was coming to the end of my PhD, I was seeing that some of these blood biomarkers that I was using in the animals were actually giving us quite a good idea about whether the drug was working or not for patients. And I was in quite a lucky position that I was working in the building where the clinicians who were running the study had their offices. So I was able to sort of corner them in the hallways and barge into their offices and say, look, I'm seeing this really interesting thing. Can we try it in the patients and see if we can monitor them? And they luckily were clinicians who were also very invested in research and ran a lot of research trials. So they were quite happy to have us come into the ICU and collect blood and CSF from patients, if they had an intraventricular drain. But what really sort of made that successful was the fact that there was a really excellent team of research nurses who could assist us with all the sort of on the ground work that really needed to happen to get the study up and running. So, you know, in terms of things like ethics and things, we took care of that. And but in terms of helping to recruit the participants, identifying participants as they came into the hospital, explaining the study to the families when we weren't there, things like that, we really relied quite heavily on the research nurses who were just fabulous. And I don't know that we would have been able to do those kind of studies without them. So I was lucky to, as I had just started this work, as I say, I was still doing my PhD, and an opportunity for a fellowship came up to do a research project within that Alfred Hospital space. So I applied for that and got that. So that gave me funding for 1 year to focus solely on this sort of translational biomarker study, which was really excellent. So we were taking blood samples. Participants were enrolled in the study on the day of their injury, and we were taking blood samples every day for 7 days. So we're in the ICU every day taking the blood samples. And that worked out really well. But I was sort of thinking, well, do I want to do this, or do I want to go overseas and or go elsewhere and do a postdoc, which was what I had initially thought that I would do straight after finishing my PhD. But it actually worked out really well to get this fellowship and stay for a year and do this kind of interesting study. And the sort of what came out of that was we found that the study actually wasn't, the dosing schedule of the study was not appropriate to have a therapeutic efficacy. So we've published now several papers on that, which then sort of helps to inform future clinical studies where, you know, we can tell from our pharmacokinetics work that they need to give higher doses and more frequent doses of the drug. So I had funding for that for 1 year, and the study was sort of coming to an end at that time. So after I wrapped that up, I, again, driven by curiosity, was really interested in military brain injury, and in particular blast brain injury, which sort of happens when people are not only in a war zone, but often in a war zone when, you know, they're driving over a roadside bomb or something like that, but also through gunfire, firing rocket launchers, that kind of thing in training as well. So I had the opportunity to go to Canada and work in a new program that was being set up by Ebola Chernak, who's sort of the guru of blast injury research in the world. And she was setting up a new sort of dual clinical and preclinical laboratory where the clinical lab would be embedded with the Canadian military, and the preclinical work would be embedded with the research arm of the Canadian military. So I was able to do clinical and preclinical research, and that was really eye-opening for me to see how translational things can be really in the right environment. So one of the things that really happened, especially in the preclinical space there, where we were modeling blast injury, was that we would meet with the medics and we would meet with the neurosurgeons and all of the sort of military medical leadership and say, what are the issues that you're facing and what would really help? And so we did that not only for head injury, but I helped to develop things like, or worked on things like wound healing patches and things like that, that could be translated very rapidly. So we would have the medics come in and we would train them in the use of these kinds of things. And then they would go back out to their units and could use them when they were in the war zone. And yeah, so there were a lot of opportunities in terms of translation. The military is obviously very invested in their own people. So in those kinds of environments, things can be translated very quickly, as long as there's efficacy for them. So in the brain injury space, we were translating things like, we had a big study looking at psychological resilience factors. And so we started a study looking at psychological readiness for deployment and how that was impacted by history of head injury, or whether people had a head injury when they were deployed. And so those are things that they're kind of continuing on with today, looking at things like saliva biomarkers, doing cognitive testing. So we started off this testing in a small group of people as they were going for deployment, and then sort of ramped that up for more and more people. And then now they do that for when they have new recruits joining, they do this kind of testing as a standard thing when they have people come in to join their units. And then after I was so that was in Canada, and after I'd been there for a couple of years, I started to get quite interested in MRI, because we were doing some MRI research, and I kind of was really interested in what are the sort of biological drivers of what we're seeing on MRI, because it seemed like to me at the time, the people who do the MR research tend to be MR physicists or computer science people, and the sort of end users, or there didn't seem to be a lot of marriage between the people who understand the brain science and the people who are doing the MR research. So a lot of the kind of research that was coming out didn't seem to be kind of answering questions about biological significance of what they were seeing on MR scans. So I came back to Australia and I joined a lab at the University of Sydney that was a really sort of high-tech MRI lab that had one of the MR manufacturers, scientists embedded within the lab to test out all the new tech and things like that. So that was a really steep kind of learning curve to me, to be from having done sort of very basic MRI stuff to developing protocols and sort of understanding how that worked. But we were in a really good position there, where I was working for a radiologist who was on staff at several different imaging facilities around Sydney and also around Australia. So we were able to recruit lots of patients and test out some of our MR technologies and some of our newer sort of techniques, which at that stage were kind of research only in larger populations. And so I was predominantly working in brain injury and in depression at that stage, but I also sort of started expanding some of the tests we were doing to other white matter diseases. And we were kind of lucky enough to start working with some geneticists who had large cohorts of patients for rare diseases that they could sort of bring in who were coming anyway for their kind of regular checkups. And one of the diseases we started working on was phenylketonuria, which is an inborn error of metabolism, as Clement knows. And this is something where, which I found really interesting, because the patients who have PKU traditionally were, for a bit of context, this is a disease that they can't metabolize protein. So they can eat carbohydrates and fat, which makes the diet very difficult. But the good news for people with PKU is that it can be managed by diet. So before this was sort of discovered, people who had PKU would have very serious cognitive deficits and would die, probably wouldn't live into their 20s, and be have very serious mental impairments and cognitive impairments. But it was sort of discovered that you can test for that in babies and put them on the diet very early on, and people, it was thought until sort of the mid-90s that people could come off the diet when they were in their early teen years and go back to a regular diet and they'd be fine. But what the evidence has sort of started to show is that actually people are having more gradual cognitive impairment over time. So now the advice is to stay on the diet for life. But in speaking with this geneticist we were working with, he was saying, well, I'm sort of starting to see that people are going downhill even when they're adhering to the diet. And I don't really know why. And so we had the opportunity that we could then implement some of our MRI research that was more kind of the research only MR sequences at the time, to look at these people and follow them up over time. So what we found, and what's sort of known in the literature, that at the early stages of PKU, the brain looks pretty normal on a normal MR scan, a clinical MR. But what we were able to show is that people who are generally adherent to the diet and otherwise very healthy, we could detect in certain people that there was a subclinical change in the white matter. But I think, so we found this sort of metric that we could look at, and we could show that people who are otherwise healthy might be good candidates for trials, and might need extra monitoring and extra support because they were at risk of white matter pathology. They were already having the very early stages of white matter pathology. So we started this testing off in just a few people and expanded it to, I think, most of the patients with PKU in the eastern states. And this is sort of an ongoing scan that we're doing for people as part of their regular and routine monitoring now. So I think what was great about this, and I think what's a good sort of takeaway for me definitely, was developing the technology and coming up with this sort of imaging biomarker was great. But I think one of the things when we're researchers and really focusing on our research is that we don't understand how complicated the outputs can be for clinicians. And so one of the things we sort of sought to do was make it easier for the clinicians to understand what's the meaning of this marker. So instead of saying, a particular level or this, we said, is this a yes or no thing? And what's the kind of threshold we'd be looking at? So that we can, when we're explaining that to clinicians, they can get a report saying, oh yes, okay, this person needs monitoring or doesn't need monitoring. And I think that's been one of the big kind of takeaways for me, is that things will be much more difficult to translate and to be taken up clinically if it's too difficult to understand, it's too difficult to interpret. And I thought maybe I'll just give some kind of general tips that I've learned, some things. I think one of the things that's really stuck with me is that finding out what's important to the clinicians and the patients is it's very important, because clinicians won't, unless it's going to be useful for them and they see a benefit for it, they're not going to be really very interested in trialing things or taking things up as part of their clinical practice. And patients can also be really excellent advocates. Patients and their caregivers, if they see a benefit for it, they'll advocate for that with their clinicians as well. And I think another kind of takeaway for me is that if you make it easy on the clinicians when you approach them if you take care of the ethics, if you schedule the meetings, if you're the one, showing them the progress, if you're doing essentially all the work for it, then it'll be much easier to get them on board. If you're even things like, I think academics can be a little bit, we're a bit loose in terms of our timelines and deadlines and things like that. But I think for clinicians, it's really important to show, that you're trustworthy and you can deliver on projects and things like that. So I think that's probably the main. Oh, but actually I did have one little story about, sorry, I'm probably taking up too much time, about a sort of the difficulties in translation. So this researchers that we were working with initially on our EPO trial have, they're probably one of the best examples of excellent translational research teams. So they are clinicians who do these really excellent trials for evidence-based medicine and in sort of the traumatic brain injury space. And they did a really excellent trial, which I think they published in about 2011 on decompressive craniectomy, which was a technique for people who have severe brain injury and they have an increase in intracranial pressure, which you can't really do much about because there's only so much the brain can swell within the confines of the skull. So the thinking was, if we just take part of the skull off, we can relieve a lot of the pressure because the brain can sort of swell past that. And it seems like initially it works quite well. The patients have quicker discharge out of hospital, but this this group of researchers were wondering what the long-term outcomes were. And what they found was, yes, the patients come out of, they leave the ICU much quicker and they're discharged home much quicker. But because the brain mushrooms out of the top of the skull, we have an additional axonal injury, so the patients had a worse long-term outcome. So they published this in the New England Journal of Medicine. This is something, they've spoken about at lots of conferences. There's been lots of work trying to stop using this technique. But as I said, that was in 2011, and this is something that is still done every day in hospitals, including the hospital that this technique sort of was disproven. I think that's a great example of the sort of difficulties with translational research. And even though you can have the best evidence, sometimes people are just doing what they're doing for a short-term gain. Yeah.
Thank you, Sarah. Some really great stories there. And it's funny, as a researcher, we all think, oh yeah, we do try and make things simple and in our translation, but because we're so focused in our work and so in-depth, we forget how in-depth we are and we can lose that perspective. So thanks for reminding us of that. I'll hand you over now to Andrew, who will introduce our third speaker.
Great, thanks, Jackie. Before I do, I'd just like to remind everyone that as they think of questions that they'd like to put forward for the facilitators to ask the panel after this, just please populate the Q&A as we keep going. All right, so now I'll introduce our third speaker for today, which is Associate Professor Cedric Barty. So Associate Professor Cedric Barty is the group leader of the Laboratory for Human Neurophysiology and Genetics. His research focuses on advancing stem cell biotechnologies to understand and cure neurological disorders. And in particular, he is the inventor of BrainFizz, which is a neuronal medium broadly used to culture human neurons in vitro. So I'll now pass over to Cedric, and if you'd like to take the stage, thank you.
Thanks, Andrew. So I'll try to be short because I think you wanted to keep some time for discussions, and I didn't prepare anything very formal, but I was thinking maybe the perspective I can bring here, and I don't really know what the attendees, who they are and what they're expecting apart from being EMCR, but from my point of view, so what I can bring is how I started very much my research career in fundamental basic, neurobiology, with always a really strong motivation to go towards translation, to have an impact for people, to have to move towards translation as much as possible. So it's been a very exciting but also slow process for me to go in that direction. So I started my PhD actually at the University of Sydney, working with animal models trying to understand how the brain, process visual information. So, not translational, or not really directly translational. And but it was fantastic to learn about the scientific process and all this. And from there, I moved to a change field after my PhD to do a short postdoc back in Paris at the Pasteur Institute. It's always a question for EMCR sometimes when we finish the PhD, whether we should stay in the same field, keep the narrative, maybe we have a better chance to get fellowship if we do, or if it's okay to change field. And look, there's so many ways to be successful, it's not going to be, everyone's going to have his own story. Mine is like, I decided to change field. I had an opportunity to go to Paris and to start working in something still fundamental, working with rodent models, looking at adult neurogenesis. And but I felt like at the time, like understanding how newborn neurons can integrate adult brain would be potentially more relevant for translation, because there's a need for replacing neurons dying from neurodegenerative disorders like dementia. And so that was what attracted me to this work. Right, so I spent 2 years there and it was fantastic. But right around that time, again, it's serendipity. There was this big, discovery of iPS cells, and which got the Nobel Prize a few years after that. And so I started getting, I felt, okay, this is the thing. This is adult neurogenesis. This is, iPS. This is my opportunity to move in closer to the patients. And I wrote a fellowship at the time in Europe. So it was a Marie Curie fellowship, which I was fortunate to get, and then to move to California to continue this work there. I remember also at the time, like going to conferences and asking advice. How do I do that? How do I move from basic science to something to translation? I went to like stations, trying to talk to clinicians. There was nobody who could give me a really straight answer, to be honest. So that that was the thing. IPSs was a win for me. I'm sure there's many different ways, but as a non-clinician, that was fantastic, because now I could still apply everything I've learned as basic science and all the tools and things, but then work directly, not working with animals, and more directly working with cells from patients. And so that was good. And so something I've learned too, and might be useful, I don't know, for other EMCRs, is if some of you are like me and really want to move into having an impact for patients, I realize also that there's other way to have impact, and one of it is biotechnologies. We've heard a little bit from the previous speakers. That's very important at every stages, and that's kind of that was attractive to me, because I realized you could have an impact in a much shorter time frame if you want, to commercialize a new biotechnologies that may not have a direct impact on the patients, but can help other researchers and expedite the translation or go down the line. So that's something to keep in mind too for those who are in this situation. And so I started, so I moved back to Australia about 6 years ago now to start my own group at Summary. And since then, I've been, expanding all the preclinical work, working with clinicians to get cells from different disease, patients with different disease, and recently trying to move some of discovery we do in the bench directly into a clinical trial, which is, really my dream. So this is possible. It's long journey, but maybe that's perspective I can bring. Maybe I'll stop here and take questions.
Oh, thank you so much, Cedric, for sharing. Yeah, no, that's a really exciting story, and that's a really very grateful to hear that. So thank you. Okay, well, I think it's time now for the open floor, the Q&A discussion. So I'll hand over to Suraj to lead the way on that.
Thanks, Andrew, and thank you to all of our speakers today. So I'm Dr. Suraj Samtani from the University of New South Wales in Sydney, and I just want to thank all our speakers today for sharing your tips, because I think everyone's going to take away something from this discussion. We have one question in the Q&A, so I'm just going to start us off with that one, which is, if you are a basic researcher, when do you think is the best time to involve clinician input?
I'm happy to answer that if you want. I think, look, as early, that's a good question, and as we've heard from Sarah, like as early as possible, you should try to involve not only clinicians, but consumers, patients, everyone, who can benefit from the work you're going to do. Is going to serve you well at so many levels, having making, the early connection with clinicians is critical. If you actually have a basic science discovery that you think is potentially translatable, knowing who to talk to and having this relationship early on would be very important. And involving also the people who will be taking the medicine that you're trying to develop is very important as well.
So Clement, you're on mute.
Yeah, I agree. I mean, for our last 2 MRFF grants, they were both translating something into clinical practice. So the first one was from some mouse study that Tony Hannon at the Flurry was doing amongst mice with Huntington disease. And we had that conversation maybe 5 years before we put in this grant. I mean, we were just talking to each other, and we always have it in the back of our mind, and the opportunity didn't come up until then. And when it came up, because we had the conversation already, we've sort of chatted about it on and off, we could very quickly put it together and put it forward. The second MRFF that we just had involved engineers using voice recognition algorithms, and that was totally left field. I mean, I was just having coffee with a friend, and he was saying, look, we're doing this in cerebral palsy, which, is nothing to do with what I do. And then within the month of that, something came up that was suitable, and we quickly put something together. I think the lesson for me is you never know, and it's always fun to learn about what other people are doing anyway. So go around, talk to people, maybe 1 in 10 of these conversations ever lead to somewhere, but sometimes something just happened to be the right time and works. So yeah, that's what I think.
I definitely agree. And I think sometimes those conversations, they may not lead to anything initially, but they'll be kind of simmering in your brain, and it might be years later where you think, oh, here's an opportunity for that, or we can apply it in this different way. One thing I think, though, especially for basic researchers, which we've done in our lab, is to have kind of community conversations where we're consulting with consumers and also clinicians, and asking them what they think is important, and using that to guide sort of the direction of our basic research in particular. So, in terms of asking the right kind of questions that will, years or maybe even decades down the track, have a pathway to clinical significance, so that we're doing, sort of need-based or demand-driven, I guess, in that kind of respect, research.
Just want to follow up on your tips about consulting with clinicians as well as with people who have lived experience. So we might start off with talking about approaching clinicians, because a lot of EMCRs worry, clinicians are very busy. How do we approach them? Should we include some sort of consultation fees to attract them towards pilot grants that we might be putting together? What are your tips for that? Sarah, did you want to start as well?
Yeah, sure. I think consultation fees can be a little bit tricky. I know I have done it before in some schemes, particularly overseas and sort of through the NIH system as well. I think here in Australia, and especially for pilot funding, it can be a little bit tricky and a bit difficult to justify. But I think in terms of approaching clinicians, if you've got a clear plan and there's not much really, especially in the early stages when you're applying for grant funded, if there's not much needed from them in terms of their time invested, and sort of there's a reasonably big payoff for them in terms of being named on the grant, that's usually sufficient. And I think the main thing is just don't be shy. As an EMCR, it's always very daunting to approach someone who's senior, who's a very busy clinician. I would say just email them. If you're in a position to talk to them in person, go up and have a chat to them. They're often much more willing to chat to you and to discuss things than you think. And if they're not, then try someone else. There's lots of different people around. There's lots of different options. Yeah, and another thing as well is, I guess, if there's a more junior person that they work with, sometimes they have a little bit more time or maybe a little bit more interest in research. So reaching out to multiple people as part of the same team can sometimes be helpful.
And the other thing is, I often go to conferences that are not in my area. So, I mean, you might want to go to a clinical conference. I mean, you think, well, you know, I will go to a RNA biology conference. Almost none of my work could be presented there. But first, it's fun learning about what other people do. And second, you're sitting amongst all these people and you just have conversations and you never know where that leads to. So particularly, you know, if something's in your city, it's on anyway, a couple of days, just go.
That's also what I found, you know, like I remember thinking, well, where do I start? Where do I find these clinicians, you know? And if you're lucky, someone can introduce you, but if you're not, then I remember going on a Saturday, you know, there was this clinician meeting and usually the clinicians who are actually talking these meetings have some interest already in research. So at the end of the meeting, I went and chatted with them, you know, and that starts something, you know, and you know, it might lead to nowhere the first time, and you try again, and maybe eventually you find the right person. Or so meetings usually is a pretty good place to start if you don't have any connections already.
Just following up on that question, we were wondering what's the best way to establish expectations with clinicians. Let's say you approach someone so that you can get a healthy agreement set up for the project to be successful going forward. Would like to go first?
I don't mind to go first. I think being very clear about what your expectations are, what you're going to do, what you need from them, in terms of time, in terms of investment, in terms of all those kinds of things, being very clear up front so they can sort of make a call. Is this something that I can be part of? Is it something I can commit to? And also being clear on what you'll be doing, and asking them, you know, is there anything else you need? Can we, you know, do you need someone as a research nurse that we can try and find some money for? How can we best facilitate this to work from both sides? And just, yeah, having clear conversations and then following up by email or things like that to reiterate what those agreements are, so it's kind of formalized in some way and on paper so that it can be referred to.
Cedric or Clement, did you want to add anything?
No, I think the same thing. I think, you know, depend on the study, regardless, you should have a protocol, you know, who does what when. You may not have that finalized when you first approach someone, but you should have it by the end. So I think that needs to be very clear. And I think if it's time critical, things to be done in a particular time, then it needs to be funded. I think people sometimes assume, oh, look, the patients are always there. Can't you do this and that? But, you know, it needs to be funded. So, you know, when I work with my other colleagues who are multi-site studies, we have very clear agreements, you know, what activities is, and transparent how much, you know, it costs and how much funds we will send everyone. So I think that that's important too.
Yeah, it's not just one answer to this question, because, you know, the level of, you know, involvement for the clinician is going to depend on the project. It could be as simple as just, you know, giving some advice to, you know, being really involved in, like, you know, actually, you know, taking measure from the patients, you know, on the, you know, long-term basis and things. So I think, you know, you gotta, the way I see it is just approach it like any collaboration, you know, like, so it's got to, like Sarah and Clement say, like, just be clear, you know, of what you expect, and be, you know, respectful of the time of the person, and try to, you do as much as you can do yourself, instead of expecting the clinicians to do everything. There's actually a lot of things you can do for the clinicians to save some their time.
I might just add as well, I think obviously the funding aspect is really important, but sometimes, actually, I think a lot of the time we have these ideas, and if we can't get funding for it, we just put it in the bin. We've tried that grant round, okay, well, we'll just give up on that idea. But I think have a plan B, have a plan C. Can you do a scaled down version of the study to get a bit of pilot data so that you can then apply for more funding and just kind of have several different pathways to make it happen if you really want to and you think it's worthwhile.
Just following up on what you said about pilot studies, if a pilot study grant doesn't have enough funding to, let's say, hire a research nurse or support the clinician in that way, do you try to get in-kind support from the clinicians, or how do you get around that obstacle?
I don't know. Maybe the clinician will be better to answer this question than me. But again, it's going to depend on the study. I think different people will have very different expectations. So in a pilot study, it's very often, we're assuming that the basic researchers are contacting the clinicians, but sometimes it's the other way around. Sometimes the clinicians will contact you, and so obviously that changes a little bit the dynamics. So I think in my experience, it's always been in kind, but maybe other people have different, at least in pilot studies, but maybe other people have different experience.
Yeah, I would say, sorry, I would say maybe, in kind is probably generally my understanding as how things usually would work as well. But I would say, especially if you're just trying to get a little bit of pilot data to then, get a little bit more evidence to go for a larger grant, especially for an EMCR, see what you can do yourself. If there's you need to take blood samples, it's not that difficult to do a phlebotomy course as a researcher, take the blood samples yourself, get an iPad, go into the clinic and do the cognitive tests yourself, do the assessments if you can. If you've got, obviously the expertise and the clinicians are okay with it, then as much as you can do yourself and, be in the clinic. And that also helps them sort of get to know you and the other staff as well, to get to know you and see you around and sort of be often a bit more willing to work with you.
Great, thank you so much. And might go on to our last question now, which is involving people with lived experience. What would be your tips for doing that? And also, as we're talking about grants related to that, how do you talk about the impact of your research in the real world as well? If you could touch
on that. I don't know if somebody wants to go first. Give it a go. How do you talk about the impact of your research in the real world? Look, again, this, all the standard, ways that we are familiar with, the first one would be publication, I would say. But then, obviously there's a lot of, forums for reaching out to a broader, audience, non-scientific audience, non-clinician audience. And I think that's often very, very powerful to engage a community. Personally, for the really clinically oriented work, I've worked a lot with foundations that are really good advocate for the consumers of a specific disease, and that's been fantastic, because it requires a lot of work to really create the network of a community of patients, and these foundations are doing a fantastic job on that. So if you can connect with them and work together as a researcher, I think usually it makes things a lot easier.
Sarah?
I might add to that if I can. Foundation's absolutely a fantastic way to get involved with consumers. often they'll also have conferences as well. So going along to their patient conferences, where I think it often gives you a good idea of the other sort of areas of research that might be going on in that disease or that illness, and the kind of other challenges that they're facing and the answers that the questions that they need answers to. But yeah, being able to meet, it's a big group of consumers especially. And I think one thing that's important is we all tend to have an elevator pitch of our research for a sort of scientific audience, but having a elevator pitch, you know, a 1 or 2 minute thing that you can describe your research and how it impacts patients or could impact patients, I think is a really helpful thing. And the other thing is media, like if you've got a publication or you're doing, even if you don't yet have a publication, but you're starting a really interesting study, every university or institute has a media office. You can contact them, ask them to do a media release. You can even, you know, try and write for the conversation or something like that, contact local radio stations. They're often quite happy to have you on and have a chat. And that can often lead to a lot of direct contact with consumers as well.
The last thing I was going to add is we often get asked what our impact was when you put in a grant application, and that's usually Saturday night. So one thing I do is go to Google Scholar or go to Web of Science and look at what papers or what articles have quoted mine. And then that's actually very concrete evidence of, you know, some of your impact.
I think that's all we have time for today. I'm mindful everyone has other things going on in the next couple of minutes. So thank you everyone for attending today, and especially to our speakers. Thank you so much for sharing your tips and advice with everyone. And thank you to the ADNeT EMCR group for organizing this webinar. So we hope everyone's learned something from today, and a recording of this webinar will be available on the ADNeT YouTube channel. Thanks again, everyone, and have a great day.
Thank you. Bye.